Hello students, welcome to my channel. Today we will discuss the topic counter current mechanism. So firstly, try to understand the definition of counter current. What is this? This is the diagram of nephron. This cup shaped structure, that is the Bowman capsule. Cup shaped structure, Bowman capsule, and these bunch of capillaries in the Bowman capsule is glomerulus. So these two parts, these are known as the renal corpuscles, and then the renal tubule, PCT, loop of Henle, and DCT, collecting duct. So that is the part of nephron. So what is the meaning of counter and current? Counter means opposite and current means flow. So if we see the loop of Henle, this loop of Henle, it is the descending loop and it is ascending. So in descending loop, the flow of filtrate in the downward direction like this. And in ascending limb, it is in the upward direction. So this flow in the in the loop of Henle in opposite direction is known as counter current mechanism, and the same type of mechanism is taking place in vasa recta. So firstly understand what is vasa recta. The vasa recta, that is the extended part of efferent arteriole. What is efferent arteriole? The efferent arteriole is a capillary which is coming from the glomerulus after filtration, because firstly the main artery that is taking the blood in the kidney, that is the efferent arteriole. This efferent arteriole then open into the glomerulus, and here filtration takes place through these three layers, and finally the remaining blood that is coming from the efferent arteriole. So this efferent arteriole, the extended part of this efferent arteriole is nothing but that is the vasa recta. It is also known as the peritubular capillaries. So this vasa recta also run parallel to loop of Henle, means it is coiled on loop of Henle. So this is the descending loop of vasa recta, and it is the ascending vasa recta. So you can see here. So what we have to remember this thing, that flow of blood in the vasa recta and flow of filtrate in the loop of Henle, both in opposite direction, mean in their ascending part and in their descending part, that is in the opposite direction, and this mechanism is essential to concentrate the urine in case of mammal, and so that these mechanisms are known as counter current mechanism. So I think you understood the definition. Now let's do the definition. Counter current mechanism means a type of mechanism that occur in kidney which concentrate the urine, that is counter current mechanism. Now the question arises, why there is the need to concentrate the urine? Because the human beings, the mammals, they are the terrestrial organisms, and the season and the amount of water in the body that not remain same all the time. Sometime there is the dehydration condition, means very less amount of water present in our body fluid. Sometime hydration is there. So at the time of dehydration, to maintain the amount of water in the body fluid, there is the requirement to produce concentrated urine. Concentrated urine कैसा? जिस यूरिन में जो वाटर है वो बहुत ही लेस अमाउंट में हो, और वाटर लेस अमाउंट में रिक्वायरमेंट क्यों हुई? क्योंकि वाटर हमारी बॉडी को चाहिए. तो इफ़ यू कैन कंपेयर विद द सीजन during the summer season and the winter season, I think you can tell this answer now, where In which season there is the requirement of counter current mechanism? You can say in summer season. Why? Because in the summer season we are in the dehydration situation, because due to the sweating most of the water from our body that is removed. So during summer season the kidney have to maintain the amount of the water in our body, and that is done through counter current mechanism. So you understood the definition also, you understood why there is the need of counter current mechanism. Before this, one thing also you have to remember, or you have to learn. First the cortical nephron, and second the juxtamedullary nephron. There are the two types of nephron. First are the cortical nephron. Cortical word, that is very clear from the name, means they having the very short loop of Henle, and the very short loop of Henle is extended in the medulla part of the kidney. So this nephron, that is the cortical nephron, because very short part of loop of Henle that is extended in the medulla part. And second, that is juxtamedullary nephron. In case of juxtamedullary, they having a very long loop of Henle, and that extended up to the medulla part of the kidney. So the name is very clear, juxtamedullary nephron. So there is the difference. If more concentrated urine is produced, or we have to do the counter current, then main role of these juxtamedullary nephron is there, because they have the very long loop of Henle. And if the loop of Henle is long, then there will be the more amount of water that is reabsorbed from the loop of Henle. After that, one term is also here, that is the osmolarity, means everywhere the values are written 300, 600, 900 osmol per liter. So what is the meaning of this? The meaning of this is the concentration gradient. ओस्मोलैरिटी जो है उसका मतलब होता है कंसंट्रेशन ग्रेडियंट, मीन्स जो सॉल्यूट्स हैं, जो आयंस हैं इनके अंदर, लूप ऑफ हेनले के अंदर या इंटरस्टिशियल फ्लूइड के अंदर जो आयंस हैं, जो सॉल्यूट हैं, उनकी कंसंट्रेशन को हम बोलेंगे ओस्मोलैरिटी. सो दिस ओस्मोलैरिटी इज सॉल्यूट कंसंट्रेशन पर लीटर. सो वी कैन से 300 osmol per liter, the amount of the solute. So you have to understand this thing also, the osmolarity. Now this counter current mechanism. So this counter current mechanism is occurring both in case of loop of Henle and also in case of vasa recta, because in loop of Henle also the flow of the filtrate in opposite direction, and also in vasa recta the flow of filtrate in opposite direction. So this mechanism of concentration of filtrate, that is divided into three parts. The first, that we can say the counter current in case of loop of Henle. So first, that is counter current in loop of Henle. Second, counter current exchange, counter current exchange system in vasa recta. And third is urea recycling. So all these 3 points we have to understand in this topic: counter current in loop of Henle, counter current exchange in vasa recta, and urea recycling. So firstly, counter current in loop of Henle. let's revise the topic functions of tubule. In the topic functions of tubule, I told you that the descending loop of Henle, that is more permeable to water but less permeable to ions, and the ascending loop of Henle, that is more permeable to ions but less permeable to water. So due to this change in the osmolarity, there will be the high reabsorption takes place in the descending part of loop of Henle, and no reabsorption of water takes place in the ascending part of loop of Henle. So this mechanism is the counter current in loop of Henle. Now due to this, what happened in case of vasa recta? So if you see the location of vasa recta, that is the descending vasa recta. This is descending, but here it is ascending loop of Henle. Ascending loop of Henle. So here what happened? From this ascending loop of Henle, because it is permeable to ions, so NaCl that is reabsorbed from this ascending loop of Henle, and this NaCl that is exchanged, that will not remain in the interstitial fluid, that is exchanged by this vasa recta. And which part of vasa recta? That is the descending. So we can say the ascending loop of Henle exchange NaCl with descending vasa recta. So this NaCl then come in the descending vasa recta, and then from the ascending vasa recta, this NaCl again come in the interstitial fluid. So by this, maintaining the osmolarity in the interstitial fluid as well as in the tubule, as well as in the loop of Henle, as well as in the vasa recta. So why we are calling it the counter current exchange in vasa recta? Because from the loop of Henle, when NaCl that is reabsorbed, then it will not remain in the interstitial fluid, but it come into the descending limb of vasa recta, and from the ascending limb of vasa recta, then again this NaCl come in the interstitial fluid. So by this method, the osmolarity or the osmotic concentration that is maintained. And what is the amount if we compare in this cortex part? That is 300 osmol per liter. In this middle part, this middle is the medulla, and the inner, that is the inner medulla, and in the inner medulla the osmolarity that is more. So due to this difference in the osmolarity, there will be the exchange of the ions, and this exchange takes place from loop of Henle to the vasa recta and also from the interstitial fluid. So this mechanism is known as counter current exchange. So here also I drawn the diagram of vasa recta. So if it is going in the downward direction, that is the descending vasa recta, upward direction ascending. So this descending, that is taking NaCl from this ascending loop of Henle and giving NaCl, that ascending vasa recta giving the NaCl to the interstitial fluid. So like this, the exchange of water also takes place, because here the reabsorption of water that takes place by the descending loop of Henle, and here again the vasa recta also take the water. So both the exchange of the water and the exchange of the ions from loop of Henle to vasa recta takes place, and this is nothing but that is the counter current in exchange in vasa recta. And by this what happen now, more amount of, because maintenance of this osmolarity, then finally from this collecting duct water is more amount of water, you can see the arrow here, more amount of water that is reabsorbed. And if more amount of water is reabsorbed, then the final urine that is more concentrated. So more concentrated urine is produced, means very less amount of water is present in this urine. And why this happen? Because by maintaining the osmolarity. And how this osmolarity is maintained? This is maintained by the counter current mechanism both in the loop of Henle and the vasa recta, because both the loop of Henle and vasa recta, they are in the close proximity, and the counter current mechanism in case of vasa recta and in the loop of Henle help in maintaining the osmolarity from 300 osmol per liter to 1200 osmol, because in the cortex region that is the 300 osmol per liter, but in the inner medulla part it is 1200 osmol per liter. Then again when this loop of Henle, ascending loop of Henle, when it is coming in the cortex region, again this osmolarity is changing. And by the changing of this osmolarity, there will be the reabsorption of water takes place from the collecting duct, and finally what type of urine is produced? The concentrated urine is produced. Next thing is urea recycling. In the very starting, when you have drawn the diagram of the nephron, the simple diagram, so at this place you are showing these two arrows, and these two arrows are the urea recycling. For the urea recycling means कि जो यूरिया था वो हमारा यहाँ से, वैसे तो हमने जो यूरिया दैट इज़ द वेस्ट, और वो वेस्ट जो है हमारे ब्लड से रिमूव होके इस ट्यूबुलर पार्ट में आया है, और फाइनली इसको हमारी बॉडी से क्या होना चाहिए था? बाहर निकलना चाहिए था। लेकिन क्या होगा? स्मॉल अमाउंट ऑफ यूरिया दैट इज रीएब्जॉर्ब्ड बाय द मेड्यूला पार्ट, एंड दिस मेड्यूलरी इंटरस्टिशियल मेड्यूलरी पार्ट exchange some urea to the ascending loop of Henle. So there will be the exchanging of this urea takes place, and recycling, small amount of urea also present in this interstitial part, and some is secreted in this ascending loop of Henle. So it is the urea recycling. So all these three mechanism, counter current in the loop of Henle, counter current exchange in vasa recta, and the urea recycling, all these mechanism help to maintain the concentrated urine, or help to maintain the amount of water present in our body. And this mechanism is required for the terrestrial organism, those who have a very less amount of water. So during the different season this mechanism is maintained, and this mechanism is maintained by the kidney, but not by the kidney, it is maintained by the nephron, because the nephron have the vasa recta, the loop of Henle, these two tubules are there, capillaries and the tubules, and they maintain the counter current mechanism. So if you understood, Please like my video, share and subscribe my channel. Thank you.